Alright guys, welcome back to Underground Science, and in this video, as you guys can see, we'll be talking about the transition phase of glycolysis. Alright, and if you guys haven't watched my glycolysis part 1 and 2 video, you guys should go ahead and do that real quick. It's not quick, it's kind of long, so hopefully you guys don't get bored and actually understand glycolysis, and then this video is going to make much more sense. Alright, so go ahead and go check those videos out. I'll put a card above for that. Alright, but so now that we're done with discussing that, let's go ahead and before we start, and this is just going to be the overview. Alright, in a future video, I'll make a more detailed version of this. But for right now, let's just figure out what's getting made in the transition phase. Alright, so like the main goal in this transition phase of glycolysis is to synthesize acetyl coenzyme A. Alright, so let's go and write that in red. Acetyl coenzyme A. Right, this is the main goal for this step because acetyl coenzyme A will then go to the Krebs cycle, and then in the Krebs cycle it's going to go and join with many molecules like oxaloacetate, but we'll talk about Krebs cycle. We'll talk about the Krebs cycle later in a future video, but what it's going to do is it's important to synthesize acetyl coenzyme A because then it's going to go to the Krebs cycle, and in the Krebs cycle you're going to make important electron carriers for the electron transport chain, and then that's going to lead to having ATP or energy, all right? And our cells need energy, all right, in order to function. So that's the purpose of the transition phase of glycolysis, to make this acetyl coenzyme A, and we'll see how, okay? So let's go and get started. If we remember that at the end of glycolysis, at the end of the part 2 video, while we were discussing how we made the 2 pyruvates, right? That's where we ended it. So the 2 pyruvates, so I did draw it like this. So let's go and just be consistent, draw like that again. Just try and draw us like a 3 carbons or whatever, right? Instead of drawing like all the chemical structure of each molecule, it's going to be way too long. So let's just represent it like this. Okay, so this is going to be our pyruvate. And remember, we started off with a 6-carbon glucose, so that means we have to end with a 2-3 carbon structure, which is 2 pyruvates, which is what happens. So remember, there's 2 of these. All right, so this is where we ended, and we said that at the end of pyruvate, I think I mentioned last video. If I didn't, I'm mentioning it. Let's just mention it right now. So when the pyruvate's done, right, we get our 2 pyruvates. If there's oxygen, then the pyruvate will then go to the mitochondria of the cell. So it's going to go to the mitochondria, the matrix of the mitochondria in particular. So let me go and write that. Matrix of the, or you know what, it just actually called a mitochondrial matrix. So let's just go and write it down like that. All right, so mitochondrial matrix. There you go. So that's the inside of the mitochondria, right? Inside the so we have the inner mitochondrial membrane, or the cristae, and then inside that we have the mitochondrial matrix. Okay, so if there's oxygen, so let's write that down, aerobic conditions. If there's aerobic conditions, the pyruvate, or the pyruvates, because there's 2, will go into the mitochondrial matrix, and then we'll start the transition phase, which we're going to discuss in this video, and then it'll go into the Krebs cycle electron transport chain, right, on the cristae. But if there is oxygen. Now in the last video I mentioned shortly that if there wasn't oxygen, we would make lactic acid or lactate, and that would go to liver for energy, right? So let's just go and recap that real quick, just a quick refresher. So what's going to happen is, remember in the let's go and write it down right here. Remember, if you guys remember in the glycolysis videos, right, we said that there was a step that went from glyceraldehyde, it's kind of a long name, so glyceraldehyde 3-phosphate, right, the phosphate's on the 3rd carbon, and we went from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. So let me go and write that down. 1,3-bisphospho. Should just abbreviated these, but it's fine. I guess it's good to know the names. So it went from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and if you guys remember, we had an enzyme involved in this step, and that enzyme was called glyceraldehyde 3-phosphate dehydrogenase. All right, so glyceraldehyde, it's an aldehyde, 3-phosphate dehydrogenase. All right, and so glyceraldehyde 3-phosphate dehydrogenase catalyzed a step between glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and then it also reduced an NAD plus molecule, right? So in this step, we also saw an NAD plus molecule. Let me go and write it all in caps. NAD plus get reduced, which means gain electrons, and turn into an NADH molecule. All right, so this is where we talk about what happens when we make those two pyruvates, and then what happens when we're in anaerobic conditions, like, if you're on like if you're doing like squats or the bench press or something, you're repping it out, you start to feel that burn. Well, you start to feel that burn because now the amount of oxygen is going less, right? We're becoming in an anaerobic state. So that's why when our cells become in an anaerobic state, from pyruvate, we don't transfer that pyruvate into our mitochondria. We straight go from pyruvate to lactic acid for energy, right? For quick energy. So we can go to liver, and the lactate can get converted into ATP or glucose. So here's what happens, right? So the pyruvate, instead of going to the mitochondria, now we're in an anaerobic state, all right? Because we're lifting weights, trying to get gains. Okay, so anaerobic state. So this is so what I'm about to write here is going to be for anaerobic. We already went through aerobic. I mean, we haven't. That's the topic of this video. I'm just recapping right now. So pyruvate to anaerobic state, and what's going to happen is that these NADHs right here, right? Remember, there's two NADHs here. I'm only showing one, but there's actually two because dihydroxyacetone phosphate gets converted to glyceraldehyde 3-phosphate also via triose phosphate isomerase. Remember from the last video, from the glycolysis videos. So there's actually two NADHs here, and these two NADHs are reducing agents, okay, because they reduce these pyruvate molecules right here. Okay, when it's in anaerobic condition, these two NADHs come over here and drop off their electrons, and these electrons combine with the pyruvates, causing the pyruvates to be reduced, and in return, these NADHs get oxidized. So in return, what happens is that these NADHs now come here, and they drop off their electrons, they reduce the crap out of pyruvate, right? And then they turn into NAD pluses. All right, and pyruvate gains electrons, all right, because it gets reduced. That's what gaining electrons means. So pyruvate gains electrons, and NADH loses electrons because it's reducing pyruvate, or it's getting oxidized. Okay, so now when that happens, we have an enzyme that helps convert this reduced form of pyruvate into lactic acid. All right, so what's going to happen is this reduced form of pyruvate is going to come here, right? Remember, we're not in the mitochondria because this is anaerobic, and it's going to get converted into lactic acid, all right? And how? Because of our friend lactate dehydrogenase, all right? Lactate dehydrogenase, all right? And then this lactic acid can then dissociate, right, into lactate and an hydrogen ion, and then this lactate can go to the liver, and then when it goes to the liver can use that lactate and convert it into energy or glucose. All right. So that's what happens in an anaerobic condition. But this video, we're talking about what happens in an aerobic condition, right? So once we're done with making our pyruvates, and if it's aerobic, we're gonna go ahead and go to the mitochondrial matrix and get that transition phase done. Okay, so this is just gonna be a basic overview of it. We're not gonna go into the enzyme names and the enzyme complexes and whatnot, but we're just gonna focus on what's overall being made. Okay, so let's say this is let's say we're in our mitochondrial matrix now. All right, mitochondrial matrix. All right, so we're here now, inside. All right, so what happens now, let's draw our 3-carbon structure. All right, so for right now, I'm just drawing 1 pyruvate, but keep in mind there's 2 pyruvates, which means everything that's going to get produced in this transition phase of glycolysis will be 2 of each. But since I'm only showing 1 pyruvate, I'm only going to write down 1 of each, but at the end we'll go over the steps and write down 2 for each, because that's what's getting produced. All right, so we have our pyruvate molecule, our 3-carbon structure, right? And what's going to happen is that we're going to meet a huge 3-enzyme complex, which again, we're not going to go over in this video. It's going to be a future video, but pretty much what's going to happen is that one of these carbons in pyruvate is going to get broken off and is going to leave as CO2, so as a waste product that we're going to breathe out, right? So what's going to be left after this is a 2-carbon acetyl group. Okay, so 2-carbon acetyl group. And remember, in the beginning of this video, we said the goal was to produce acetyl coenzyme A. So this is our acetyl group. So we have, I guess you could say, oops, that's not your spot. I guess you could say we have half of what we need, right? We just need a coenzyme group now, a coenzyme A group. So we're going to get that by the second enzyme complex in particular, but for right now, let's just say that what happens is that a coenzyme A will come here and bind to the acetyl group. All right, so this coenzyme A group right here will come and bind here. And overall, now you guys probably guessed it. If an acetyl group is there, a coenzyme A binds, we're going to have acetyl coenzyme A. Okay, and that was the whole goal. So we're going to have a 2-carbon acetyl group, right? Acetyl group. And then we're going to join our coenzyme A group, making acetyl coenzyme A. Okay, so let's go and draw that coenzyme A group here. All right. So right there, bam, right? We've got our main goal, which is acetyl coenzyme A. Now, what also happens, right, is that we produce an NADH molecule, right, for every pyruvate. So right now we're only drawing 1 pyruvate. So what happens is that in one of the 3 enzyme complexes, right, in particular the last enzyme complex, what happens is we reduce a NAD plus molecule and turn it into an NADH molecule. All right, so let's go and write that down. So we produce 1 NADH molecule, all right, in the transition phase of glycolysis. And that's it, all right? That's pretty much it. So again, but this isn't, technically all of this isn't right, because remember, we drew 1 pyruvate coming in, but there's actually 2 pyruvates at the end of glycolysis. So that means if there's 2 pyruvates, we're not going to let go of 1 carbon dioxide molecule, we're going to let go of 2 carbon dioxide molecules, right? We're not going to make 1 acetyl coenzyme A molecule, we're going to make 2 acetyl coenzyme A molecules, right? And you guys probably guessed it, we're not going to make 1 NADH molecule. Oops, sorry, I was pointing here. This is the actual acetyl coenzyme A joined, all right? So let me just go ahead and fix that real quick. Right here. These are the 2 acetyl coenzyme A molecules we're going to make because of the 2 pyruvates. Let me draw that better. All right, and lastly, we're not going to make 1 NADH, but we're going to make 2 NADHs because we have 2 pyruvates. All right, and this NADH right here, these NADHs will then help us produce energy, right? This will help us produce energy in the electron transport chain. So let me just go and write that down. Overall, it's going to help us produce energy. All right, and that's pretty much it, guys, right? That's the transition phase. We'll go over it in more detail in a future video, but for right now, that's good enough to know, right? 2 NADHs, 2 acetyl coenzyme A's, 2 waste products of carbon dioxide, and we make 2 NADHs. I think I already said that, all right? So guys, that ends the video right there on the overview of the transition phase of glycolysis. In my future videos, I'll be making, in the future, I'll be making videos about topics related to more biochem, physics, and chem. So hopefully you guys tune in. Don't forget to hit that notification bell so you know when I'm publishing my videos. And don't forget to subscribe and like, all right? So see you guys next time, and stay safe.